Hi, I'm Roger Bull, Head of Operations at the National Biodiversity Cryobank, which you can see behind me here at the Canadian Museum of Nature's Research and Collections building. I like to think of the cryobank as a DNA library on the dark side of the moon. DNA molecules are the instruction books for life. We preserve the information in these molecules at the temperature of the dark side of the moon. That's colder than minus 160 degrees Celsius. The books in our library are bar-coded vials of frozen plant and animal tissues, good sources of DNA. We have thousands of these. They are digitally databased and stored in freezers. This allows for easy retrieval when we receive loan requests from researchers. The cryobank is home to many kinds of samples, each with an interesting story. Let's take a closer look at these little frozen multicolored vials, our collection's polar popsicles. Eating them is not recommended. These are Arctic microbe samples, and some may be toxic. The Arctic is vast, with few roads. Residents and visitors travel between communities by air. From an airplane, you can see a landscape dominated by water features, ponds, lakes, rivers, wetlands, and the ocean. These are habitats for the biodiversity we usually associate with the Arctic: polar bears, eider ducks, and Arctic char, species both ecologically and culturally important. These animals are only the tip of the Arctic biodiversity iceberg. Since the theory of evolution was proposed, scientists have been drawing trees of life to represent the diversity of species and the relationships between them. With advances in DNA sequencing technology, scientists are discovering most biological diversity on Earth is bacteria and other microbes. Non-microbial life, including all plants and animals, fits on a small branch of this updated tree. Laval University researchers Connie Lovejoy and Warwick Vincent have dedicated their careers to studying the diversity of microbes in Arctic waters. Much of this microbial diversity is invisible to the human eye. Dr. Lovejoy studies marine microbes. In 1 liter of Arctic ocean water, there are over 100 million microscopic cells, including bacteria and larger microbes like phytoplankton. Seawater may look clear to our eyes, but it's teeming with life. We've come to learn that bacteria and other microbes are essential to complex food webs. Bacteria keep the show on the road at the base of the food web by recycling nutrients and feeding larger microbes, which in turn feed the rest of the web. Microbes also show up in the Arctic as mats that cover rocks in freshwater environments. These are a focus of Dr. Vincent's research. These mats are surprisingly complex, a community of many different types of cohabiting microbes, including bacteria, fungi, and viruses. Cyanobacteria, such as these species seen under a microscope, are the dominant organisms in microbial mats, giving the mats their color and structure. Cyanobacteria are photosynthetic, using sunlight to make their own food. Microbial mats provide habitats for other organisms, such as this chironomid larva that you can see crawling around. This larva will grow into an adult fly unless it gets eaten. Chironomid larvae are the preferred food of Arctic char. Here is Debbie Ikaluk from Kausuituk, or Resolute Bay. She was assisting Dr. Vincent's research team when her keen eyes spotted small Arctic char in one of the lakes being sampled. Char are good eating and important to northern communities. Warwick Vincent and Connie Lovejoy have collected and isolated cyanobacteria from many polar environments. These isolated cyanobacteria are grown in flasks, making this a living collection of Arctic microbes housed at Laval University. And here we are, back to our polar popsicles, frozen vials of cyanobacteria archived in the cryobank. They are colorful because each species has its own palette of pigments for capturing sunlight. Warwick Vincent has also deposited microbial mat samples into the cryobank. Instead of single cyanobacteria species, these less colorful vials contain a rich community of diverse microbes. But why cryopreserve these samples? The Arctic is warming much faster than the rest of the planet. Large ice shelves have collapsed. Lakes that used to be ice-covered all year now have open water in summer. Others have completely dried up. Permafrost is thawing. In short, ecosystems are disappearing. Archiving microbe samples in the museum's cryobank is one way of preserving this rapidly changing biological richness. Like books in a library, these samples are a resource for scientists to better understand these microscopic organisms.